Becky's in here with me. All right, so we're in my lab, and I'm going to tell you a little bit about the work that we do in the next 8 minutes or so. And it's going to look like this as we go. People are going to be masked. I wish I had a wide screen. Maybe I'll just walk you around a little bit. Nice mic. Can you still hear me? Okay. So I am in the Applied Biomechanics lab right now. And the mission of our lab is sort of twofold. We try to leverage a lot of fancy technology I'll walk around and show you to understand the mechanisms by which people lose their mobility, their walking ability as they grow older, or if they have a neurodegenerative disease like multiple sclerosis, or a joint pathology like knee joint surgery. And then the second thing we try to do is we try to mitigate that. By leveraging the mechanistic understanding that we have, we try to come up with novel ways to engineer new training paradigms, new devices, or new systems to promote. Hi, thank you for the opportunity to speak. Oh, thanks, Anna. We want to preserve walking ability, and we want to prevent falls. One thing we have in the lab that's behind me is an immersive virtual reality environment. I'm going to walk over and turn off the lights and turn that on and then tell you about that. Actually, Becky, could you turn off the lights for us? And this computer's gone to sleep. Okay. So it's hard to see. I wish you were here. It turns out that as you get older, or if you have a neurodegenerative disease like multiple sclerosis, the sensors you rely on the most for balance, go bad. The sensors under your feet that tell your brain how to make corrective motor adjustments from one step to the next. And it turns out the brain in an older adult or someone with neurodegenerative disease, the vision, visual feedback, the brain says, I need something, give me anything at all. And our lab basically uses vision to trick the brain into thinking you're experiencing a fall so that we can do something about it. Diagnostic tests for people with multiple sclerosis, training paradigms to make balance better in your older relatives. How do we measure that? You want to get those lights again? Around the room, we have a 16-camera motion capture system. All along the wall, you'll see cameras that can measure the three-dimensional position of people as they move. Now you're going to get dizzy. And it might look like this on the screen. Is that hard to see? How's that? Yeah. So we can measure that. We have sensors that measure electromyographic activity of muscles. So we can look inside the muscle and find out how your brain is controlling that muscle. We have, as I'll walk over and show you in a moment, a lot of ultrasound imaging equipment. The ultrasound that we do, we're not on the development side. You're going to see a lot of people that are developing ultrasound technologies, especially when we get downstairs onto the 9th floor of our building. We're very much interested in leveraging that technology to make meaningful insights as to what's happening under the surface. How do muscles and tendons interact during the activities that people have problems with, like walking? So what you'll see in a moment, 10 minutes of lab, what you'll see in a moment is the fact that we've used 3D printed orthotics to house ultrasound transducers that you can wear when you walk, when you run, when you jump, right? So we can look under the hood at the muscles and tendons powering that movement, and we can figure out how does that change as we get older. We can figure out, understanding one major project in the lab, understanding how muscles and tendons interact as we get older is critical if we're going to build and design and optimize wearable assistive technologies to make walking easier for your older relatives and community members. The idea that a passive elastic exoskeleton to assist movement needs to be optimized to the biological tissues under the surface so that you can tune the device biology interaction. Let me walk you over there. For example, we can 3D print with soft materials a housing into which we can place ultrasound transducers. We're doing our best, folks. Into which we can place ultrasound transducers, and then people would walk with that. Others that we work with the P&O department, the prosthetics and orthotics department at the university. Becky, what am I missing? Oh, metabolics. One thing we've grown really interested in is linking the mechanics of movement to the metabolics of movement. How difficult it is for you to walk, how much oxygen you consume to perform an activity is governed by the mechanics of the way you move. Unfortunately, older adults and people following stroke consume oxygen at a rate of about 30% faster than you do, even if they're walking at the same speed. And the determinants of that increase are really not well understood. And so what we're trying to do is link muscle level metabolic energy costs, for example, with modeling and bioenergetic simulation, with things that we can measure at the whole body level, like rates of oxygen consumption. I have about 2 minutes to make it to the other lab. If there's one question, if you want to pop it up into the chat, I'd be happy to answer. It'll have to be the first one I see if there are any. You can also put, Becky can field any changes we need to make live to how we're doing this. If there's no questions, or we'll see them later. Yeah, so question was about human subjects. Almost all of our research involves humans and patients, whether they're coming from the hospital and walking over, whether they're coming from the community. We are making measurements. Unlike a lot of biomedical engineering labs, I like to work with live humans and interact with people and see the people in front of me that I'm going to make an immediate impact on through the research. And I think a lot of people that gravitate to the work in our lab have that same feeling. All right, I'm going to leave this lab. Let's go check out some others. It is not a Vicon system, it's motion analysis, but they all do about the same thing. See you soon.
This is Jinhee Lee from Dr. Koji Sodo's laboratory. Yeah, then let me show our laboratory and introduce our research topic. Yeah.
And is that okay? Can everybody hear okay here? Good.
Okay, so let me start about our research topic. So our research purpose or ultimate goal is to make theranostic system, which involve the monitoring of the patient's condition and also the administration of the drug. So which means to administrate the drug, we have to know the condition of patients, so that by using these two steps, we can give the patients very suitable and very good treatment. Yeah, and our main focus is this to make some medical device to monitor the patient health. Yeah, then often we start from some molecular engineering. But anyway, so let me introduce about our target. This is just example, but for example, we are now trying to make a medical device for the diabetes patients, where we have to measure the glucose, or sometimes have to measure the insulin. And for another example is just mental disorder. So currently we have no quantitative monitoring method for the mental disorder, but by monitoring some biomarker in our body, especially in the blood, we can quantitatively monitor the patient's condition. Then we can administrate appropriate amount of medicine to the patients. Yeah, and to do so, we have to make some protein or ligand which can capture and detect our biomarker in our body. So we often start from this step, which is, for example, engineering of the antibody, which is kind of the protein which can bind to some protein or other small molecule. so that we can specifically know the presence of these biomarker in our body. Then once we get this kind of ligand, we go to the sensor development, which is mainly we are focusing on electrochemical sensor. Yeah, here we have some specialist of the electrochemistry, so maybe he can explain about what the electrochemistry is.
Yeah, to add to this, so as Jinhee said, we developed a molecular recognition element which selectively binds to a biomarker that is representative of a disease state. And the methods we transduce that signal, that binding signal, is through electrochemistry, which is basically the measuring of the transfer of electrons over an interface. So in our case, we use a gold electrode polymerized interface, and we measure the change of, say, currents for enzymes where they produce electrons, terms of for impedance, in terms of resistance, and voltammetric will measure, for example, changes of current over a range of voltages. And we use these transductive methods to then back calculate how much of a certain biomarker is within the given sample.
Yeah, thank you very much, David. And fortunately, also we have 2 undergraduate students who is working more than 2 years for our lab. Then they can introduce about their work. Yeah,
that'd be great.
So yeah, as Jinhee and David kind of introduced, they talked about a lot of the science, but just as from an undergraduate perspective, I started working this lab in sophomore year, and it's definitely something that I would encourage a lot of students to partake in, just because you learn a lot in your classes and whatnot, but to see actual practical applications is something that really helps solidify your learning. And here I see a lot with the engineering. I work with as Jinhee said, for introducing my research, I work a lot on enzyme engineering for the sensing aspect of the research here, but also I do work on like device development, and that's where you see a lot of the engineering aspects really solidify. So I definitely recommend everyone to find a place in a research laboratory in this time. So Ty, you also want to introduce?
Hello, my name is Ty. I'm also an undergraduate here. And like just echoing what Karthik said, I definitely encourage you guys to all join a research laboratory if you haven't. I also work on a similar project as him where I deal with enzyme engineering, enzyme production, you know, and I'm a biochemistry major and I'm in a BME lab. So like, I mean, like you can be any major you want, but like, yeah, I definitely encourage to join a lab.
So if I could ask a question and anybody could talk, how has your research pivoted amid COVID? And do you have undergraduate research opportunities? Are you allowed to have undergraduate research opportunities right now, and including any computational projects that can be done remote?
Oh yeah, that's a good question, and that's we are trying to do. Yeah. So for example, for the engineering about our protein ligand, we have to introduce some mutation to improve the function, such as stability or specificity. Then to do so, we have to see the structure of protein and predict where to be engineered. Then we try to start this kind of project because, yeah, which can be done through some computer. And then, yeah, definitely we will do. Yeah.
That's good. Well, let me thank you again. This has been really good. You're other than my own lab where I just gave a tour, this is our first one. I'm going to go ahead and log off and thank Dr. Koji Soda's lab, and I will log in from another lab in a moment. Cool, we're still doing okay. We're in Dr. Paul Dayton's lab now, so.
Hi everyone, I'm Phil Durham. I'm a graduate student in Paul Dayton's lab. I'd like to tell you a little bit about what we do. So Paul's lab is all about ultrasound. And we've got 2 main kind of strategies. We want to use ultrasound for imaging, and we want to use ultrasound for therapy and for drug delivery. So that involves making ultrasound images better. If you've seen it, tell you what, we've got a couple of posters up over here. On the ultrasound imaging, we can add what's known as a contrast agent. In our case, it's a microbubble. Microbubbles behave non-linearly compared to tissue, and so we can actually use some math and some physics to isolate the bubble's signal. And usually they'll stay confined to the blood vessels, so we can detail these vascular maps and we can see how they change tumor vasculature in response to therapy. Over here, we're using contrast imaging to generate super resolution images with some deep learning and machine learning, if that's something that's interesting to you. We do everything from programming the ultrasound machines to post-processing the images to improve resolution. Ultrasound is kind of limited by, it's a higher order of magnitude than something like MRI, but we can improve that resolution down to the micron level. To do that, we've got a couple of engineering workstations. We have full electronics lab. We've got some wet chemistry and some fabrication stuff. We have 2 animal imaging rooms where we'll do our procedures. We also use focused ultrasound and microbubbles to do everything from targeted drug delivery to the brain, to targeted drug delivery to tumors, to immune cell recruitment to tumors to improve immunotherapy. We do that with a couple of different focused ultrasound systems. This is an example of one of our animal treatment rooms. We have basically a Xerox scanner for animals, which is the Sonneval Vega. It takes ultrasound images, but it also does focused ultrasound, which a focused ultrasound transducer looks something like this. It's a large angular array that's concave and focuses ultrasound to a point. And when I give oxygen microbubbles, nitric oxide microbubbles, I can use those for therapy. If I use perfluorocarbon microbubbles, I can use this to deliver drugs across the blood-brain barrier in a targeted manner for treatment of cancer and neurodegenerative diseases. So that's some of the animal work we're doing here. On the engineering side, again, we program a lot of our own ultrasound parameters. And this is one of our engineering labs. So Ryan's in here. Say hi, Ryan. So here, Ryan is controlling this custom-made ultrasound transducer over here. I'm on the therapy side, so I'm not one of these computer guys. But if that's something that's interesting to you, they make pictures look amazing and they make things possible that weren't before. We've got optical setups. Probably due to time, I won't take you to the other part of the lab, but we've got some space over there where we've got 3D printers. We always need things fabricated. So if that's something that you're interested in and you'd like to see it come to fruition in an actual scientific experiment, we always need prototyping. We've got a lot of applications for machine learning, a lot of coding, a lot of making stuff, and then my expertise, therapy. So if you guys want to actually get into some therapy experiments, you know, Paul's lab is a great place to grow.
So if you didn't know, Paul Dayton is the interim chair of BME. So he is our captain and leader. Something that I would be interested in knowing, and I know they would as well, and they know what question is coming because I've been asking it of everyone. Amid COVID, what type of undergraduate, to what extent have you had to pivot? Are there still opportunities for undergraduates to become available, knowing the policy about new volunteers in the lab?
Right. So on the engineering side, I think there's actually quite a bit of things to do. If you are interested in medical imaging at all, again, to go back to some deep learning stuff, we've also done some fractal analysis on some of our data in terms of improving resolution, especially in terms of localizing microbubbles. So computationally, there's actually quite a bit to do if that wouldn't not necessarily involve a trip to the lab. I'm actually not aware completely what the restrictions are on undergrads. For graduate students, we are operating under the laboratory precautions, but we've been able to move forward with research. And on the therapy side, there's always data that needs analyzed. So if you're interested in learning more about how to process biological data, how to do statistics on biological data, how to transform ultrasound images into statistically meaningful therapeutic results, those are all things that we could use a hand with in the lab. Again, from time to time, we'll need things made, and modeling is half of that battle. So if you're good at CAD and that's something that's really interesting to you, I don't know what it's like for actually doing the 3D printing yourself, but we have printers here, and modeling alone would save us quite a bit of time, and something that if you're interested in designing stuff, coming up, basically encountering a problem that you need to solve and figuring out how to model your way out of it is something that a lot of our previous undergrads have helped us with quite a bit.
All right, well, I want to thank the Dayton Lab again. I'm going to sign out from here, and I'll check in with you in a couple minutes.
First of all, hi, I'm Diana. I'm sorry that I'm your virtual guide today and not a physical guide, but you can see my collaborator Ruth can show you around. So I'm a member of the Gianmarco Pinton Lab. We are an ultrasound lab as well, and we're adjacent to the Dayton Lab. We actually collaborate on a lot of projects together. but we do not deal with therapy and drug delivery. We are a mainly imaging-oriented lab. So basically, we're trying to make really nice ultrasound images. Now, there's a lot of projects that we're working with right now. We mainly do super resolution imaging. And personally, my PhD is on transcranial super resolution imaging. So as you know, usually we can't really do ultrasound on the brain because skull, but we have developed some techniques that can give us really amazing images of the brain vasculature. And we also do that throughout the body. So deep abdominal imaging, and basically it's like having a CT angiography, if you're familiar with that, but with ultrasound, which is cheaper, more approachable, and much easier to apply clinically. Now, all of our projects are clinically translatable. We do a lot of in vitro experiments, in vivo experiments, and also pretty soon we're gonna transition some of the super resolution projects to humans. So Ruth, if you can guide them to the huge 3D scanner,
yeah,
that'd be nice. Huge 3D scanner, can't miss it. And we do ultrasound in 2D and in 3D, and we have recently, in collaboration with the Dayton Lab, gotten this, if you can point towards your left.
Diana, we're using the 3D scanner.
Yeah.
Keep going.
Okay, it's a very, very big scanner. It's like the size of half of the room. You're not missing anything. And apart from that, yes, pipette tips. And apart from that, we also do a lot of fabrication in our lab, just the Dayton Lab mentioned. We do 3D printing. We try to print all of our custom made holders for transducers, etc. And another big part of our work is simulation, thankfully, because during COVID, that's all I've been doing, basically. So Dr. Pinton has developed an amazing simulation tool for ultrasound. It's called Full Wave, and it can simulate real experiments and ultrasonic propagation as if you were doing an actual experiment. So that's a very big aspect. And we also have another project that Ruth is also working on partially, and my collaborator Sandhya. It's a shear shockwave project, which we test in live animals. And we are basically assessing the effects of shear waves on TBI, traumatic brain injury, which has been very hot recently. And last but not least,
I can show them our 2D skull.
oh, okay, yeah.
Yeah, so
And,
we're doing an experiment with a skull mold. So instead of using human tissue, we've made our own fake brain because it's a lot easier to measure the physics in, and we don't have to acquire a brain. So we've already made one today, and it's currently sitting in an ice bath. Yeah, if you want to get down here, but we use gelatin. And so all this black stuff is just Jello. And we put graphite in it to make it black. And we're going to hook it to a shaker thing, and we're basically going to shake it up and down. And we're going to simulate traumatic brain injury with it. So yeah. Okay,
we also
continue.
like pigs, but that's easier. Yeah, so that was the only demonstration we can provide today, but it's fine. And I was saying that in the past, we've had undergraduates in our lab deal with the fabrication side of things. Some have helped in the image processing side of things, but basically, I guess it could be anything as long as COVID allows us. And if you have any questions, let me know.
I wonder, we've been asking this a lot. I mean, you did a really nice job presenting as you went about how COVID has impacted your research and the research of the lab. I know a lot of our undergraduates are concerned that the opportunities that they may have otherwise had for undergraduate research simply aren't there. And my goal is to convince them if I'm trying to steer your answer in one way, convince them that those opportunities are there and that we are thinking thoughtfully about how undergraduates can be engaged in research in the department. I wonder if you could speak within the lab.
Yeah, like I'm living proof and example of how you can adapt through COVID, because actually right now I'm in the Netherlands and I've been here for months actually, and I have continued my work normally. It's just that I had to shift to our simulation side of things, and now coming back to the US, I can continue my experiments, but it's a collaborative effort. So if I need some experimental data sets, someone who's actually physically present in the lab can help me. And it's just a matter basically of managing, you know, time in lab, because we have limited space and only like 2 people per given square footage can be present. But I don't think that's gonna hinder any research opportunities in reality. Like you can definitely do stuff, especially in labs like ours where we have image processing, simulations, like so much computer work.
That's great. You mentioned working with live animals. What's your experience with that been? Are there undergraduates that get the experience of working with live animals in the work as well?
Sure, of course. If they want to, you can, of course, do the formal animal handling training. I mainly do rats and mice, and everything else I do is dead, so I don't need training. And if you have the stomach for it, like we do dead people, we do dead pigs. It depends on the experience you want to gain.
That's great. Well, I want to thank the Pintone Lab again. Thank you for doing such a nice summary. Enjoy the Netherlands. I want to.
I'll be back soon.
All right. I am going to. I told everyone that I would follow up with contact information and lab websites, so I'll do that for this lab as well.
And I'm also putting my email on the chat if anyone has any questions.
Thank you for doing that. I'm going to go ahead and sign out from this lab, and we'll be on to our next. And thank you again. We are now in Dr. Katerina Galippi's lab, and we have a guest to give us a tour of that.
Hello, I'm Kirti. I'm a third-year graduate student in the Galippi Ultrasound Lab, and we work with the ultrasound as the past 2 labs you have seen. But our specialty is with detecting tissues, tissue characteristics, and seeing how they apply to disease states and measuring that with the ultrasound. So the specific type of ultrasound we use is acoustic radiation force impulse. It's essentially where you focus sound to push on tissue, and how it responds when you push on it tells you a lot about the material characteristics. So here we have the wet lab space. We do a lot of our tests on phantoms, as well as several non-animal experiments out in this area. I'll show you the area where we do our animal experiments. But you might have seen ultrasound scanner such as this one in the hospital, and this particular model lets us do elastography applications. These transducers deliver focused ultrasound pulses into the tissue, and for this model we have phantom, which we experiment on. And with this, we are able to detect how the tissue responds when we push on it, and actually map the material characteristics. So I'm not sure if you could see on the screen, but we have a brightness mode ultrasound image, and you might be able to make out this, this small inclusion. However, in disease pathologies, even though small area of whichever part of the tissue you're looking at may appear the same, it would have different material characteristics. So if we use an RF elastography approach, we are able to map the material characteristics, and this allows us to better delineate this portion of this inclusion, which is inside this phantom. And whenever we develop scripts that we want to test in humans, we first test it out in these phantoms, which are essentially representative of the material and acoustic characteristics that we would find in tissues in humans. And while most of the work on these scanners is done by graduate students, we also have a lot of undergraduates who worked in the lab in the past, who have worked on machine learning applications, who have worked from the computer science department for bringing this technology to real-time visualizations. Usually there's a bunch of data that you need to process, which takes a long time. And we've had undergraduates from computer science who have helped make this technology work in real time. And other undergraduates have worked on projects using this technology to develop better methods of testing material characteristics, as well as analyzing the data. And if you do end up working in this lab, you have a nice view of the rest of the campus over here. And I could take you to our procedure room where we do all of our wet lab animal or animal tissue experiments. And this is like a BSL area, so you need to get training. But as far as anything with the human tissue that we want to experiment on, as well as get the material properties of, we would set it up on the motion stage and use either one of our many ultrasound machines to acquire data. This particular machine is very nice because it lets us use MATLAB, which many of you might have worked with. And using MATLAB, you're actually able to control this ultrasound transducer over here and acquire and process the images in real time. And as far as our other computational work, we have a computational design area where most of the graduate students work, which we'll show you in a bit. And here are one of our graduate students. This is Joey, and he's a second-year graduate student. And usually all of our computational work is done in this area, and where a lot of the nitty-gritty science also happens. So if y'all have any questions, you can let me know.
I had sort of two questions. The first, you mentioned MATLAB, and I know in past years when we've done this tour, students get a little bit nervous about not coming in with all of the requisite skills they would need to be successful. And so I just wondered, to what extent do your undergraduates know all of the MATLAB that they would need coming in versus how much of it is taught to them and something that the lab can provide as part of their training?
It's easy to start out with the MATLAB. We have a lot of example scripts that new students can start working with, seeing how the syntax work, how with certain data types you could end up with an output. But we also have a graduate student mentorship for anything you might need help with. And Joey could also answer a few questions.
Yeah, a lot of the programming ends up being something that you can learn on the job. We also have some undergrad CS majors that have worked with us in the past that come in and help with that as well. So,
yeah, but we've also had new rising high school students or students who are about to enter college come and work with us over the summer, and they've done a lot of phantom experiments that you don't necessarily need computational MATLAB knowledge for.
Yeah, it's a great answer. My other question just has to do with undergraduate opportunities within the pandemic. We understand that I think a lot of our students are nervous that they won't have the opportunities at their disposal that they may have had otherwise. And I'm trying to convince them that those opportunities still exist. And it's just that many of us have had to pivot in what those experiences are like. So I wonder, just amid COVID, is the Gillespie Lab looking for undergraduates, and what those experiences might look like if we're not allowed to come into the lab, for example?
Yeah, we definitely have many ideas, projects that we need.
We've been talking to Dr. Gillespie recently about setting up some proposals and sending them out. it's really just a matter of working with the individual student and figuring out what they're wanting to accomplish and what their background skills are and what they're wanting to learn, to scope out a project that would work well for them. But there are a lot of opportunities, and we are flexible and can work with the student if they're interested.
Yeah, just be a self-starter, and if you realize you don't know something, let us know, and we could help you fill the gap. So you could be, you could get what you want out of this research experience.
All right. Well, thanks to the BME Lab. Thanks to both of you for participating. Here I go again. I'm going to sign out, and I'll check in with you in a couple minutes. We're going to be back on my laptop. Your experience is probably better when I'm using my laptop, but I'm afraid my arm, if I use it on every one of these, it's just going to fall off. And then you'll see me teaching, and I won't be nearly as successful. All right. So I'm going to hand it over to our next tour person here.
Hi, everybody. My name is Jordan. I'm a third-year PhD student, and I'll be showing you the BME Lab today. So right now I am on the HPLC. We are a drug formulations lab, and so we use all of this equipment to quantify how much drug are in our implants. So kind of our bread and butter technology are what we call ISFIs. So this is a solution that we can inject into either mouse or human. We've also been all the way into monkey studies, but this can be injected, and then once it hits that aqueous environment inside the body, it turns into this polymer-based implant. I don't know if you can see that, but as this polymer degrades, the drug is slowly released in the body, and it can be released from months to all the way up to a year. So it's pretty helpful in drugs that, you know, prevent a patient from having to take a drug every day. So are we going to walk around the lab? Okay,
yeah,
cool.
Totally.
Awesome. So I showed you the HPLCs. So we also do some cell-based therapies. And so this is just part of the lab. Most people do bench work. And so you'll see a lot of people working at their bench. And yeah, I want to know what else to show. We also have this bay over here too. We have the lyophilizer, which we use to dry the implants, and then we can, you know, do some testing after that. But yeah, I think that's pretty much it.
So that's good. I wonder if you could speak to the extent in the BME Lab that undergraduates, do they play a role, and especially amid COVID, when many of our undergraduates, they're sort of maybe in the back of their minds disappointed that they won't have the opportunities available to them that they might have otherwise had. I wonder if you could speak to opportunities, maybe computational work that can be done remote. Have you been thinking about any of those types of things?
Yeah, so I don't know work specifically that would be remote. We're a pretty heavy wet lab, but we do have 2 undergraduates that are actively working in the lab. One is working with cell-based therapies, and the other one I'm not sure. I haven't seen them come in since then.
Yep.
But yeah, so there definitely is opportunity if you feel comfortable and safe.
Absolutely, that's the most important thing. Another question would be if you could just speak to, I know when you give a tour of the lab, there's a lot of fancy equipment, but a lot of people on the line here might be really interested in knowing the vision of where that equipment ultimately gets. So in terms of the mission of the lab and the type of individuals you see the work benefiting, maybe speaking to that?
So as far as like if an undergraduate were to join the lab, like what would their goals be, or
Not
what would
so
we
much,
be
but the lab
looking
as a mission of the lab and the patients that you not interact with through the research, but ultimately that, you know, the research would go on to help.
Yeah, so kind of historically we've been benefiting patients with HIV, but also with more like female type related applications such as like unintended pregnancies, STIs, things like that. So if you're interested in women's health applications, that would be an awesome way to kind of go that direction.
Yeah, very cool. So I hope everybody's appreciating the scope of research that's done in the department in BME. I've let them know that they're going to be sent a list of labs with websites and contact information. And so here's another lab doing research in biomedical engineering. Thanks so much for the tour.
Yeah, of course.
I'm going to see if there's any questions in the chat just while we're here. I admit, if you've sent questions while I've logged off and logged in, I think it refreshes. What is the name of this lab? That's a good question.
So BenHabour, it's spelled BANHABBOUR.
And if you missed that, you'll get a list after.
Yes.
I should just take this little thing off. If you have another question, feel free to send it to the chat. Becky, our host, thank you so much. If questions have come in, you'll have to just let me know. You might unmute yourself and let me know, because I don't see any of the chat. When I log out and log back in, it refreshes. Are we enjoying our afternoon so far? Some people have grown really comfortable. I see some outdoor people. One person looks like they're outdoor, but clearly they're faking it with a virtual background. All right, I'm gonna log off from this one. Thank you so much. We'll see you soon. We have an esteemed guest with us. Not that our previous guest, I'm in the same spot, so I just called our previous guest unesteemed indirectly, and she looked over at me. All of our guests have been wonderfully esteemed.
Hi, everybody. My name is Dr. Imran Rizvi. I am an assistant professor here. I recently joined the faculty. My research focuses on cancer. So I have been involved in cancer research for 30 years now, about. We get approaching that. And at UNC, I'm particularly focusing on ovarian cancer. We've made tremendous strides in improving the lives of cancer patients for many other types of diseases. Breast cancer, a lot of leukemias and lymphomas, people are surviving those when they weren't just a few years ago. Ovarian cancer, pancreatic cancer, and brain cancer remain three of the toughest cancers to treat. And my program at UNC is aimed at understanding why ovarian cancer remains one of the deadliest malignancies among women and is the deadliest gynecologic malignancy. And it has persisted that way for many, many decades. So one of the things that I work on is I take a device like this. Well, I guess you can't see it, so I'm gonna go ahead and open this one so you all can get a better look. So we take little devices like this and we put ovarian cancer cells in them. And my lab during my NCI funded work was one of the first to take ovarian cancer cells and grow them into little 3D tumors inside a device like this. And what we found was when we flowed fluid over those cells and those little tumors, they became harder to treat with chemotherapy. And so now what I'm trying to do is to figure out why that's true. Why, when they grow in this little environment, which is like the environment that the tumors will grow in inside the belly of a patient who gets advanced disease, why does that disease become harder to treat with chemotherapy like most patients receive? And that's what I do here. So my terrific postdocs and students are spending their time figuring out more sophisticated ways to model the disease in little bioengineered devices. And we're also looking at factors like environmental contaminants that make that disease more aggressive. So essentially, I'm trying to model the ways that the disease becomes aggressive in the lab so that we can find better ways of treating it in patients. All right. And so I'm going to show you some of the things that we use to do our work. So a lot of my work involves cell culture. So there's these big hoods here that allow us to grow the cells in a sterile environment. Some of you may be familiar with this, others may not. So basically we grow the cells and put them inside these devices in hoods like this. And then we create a very comfortable environment in these incubators for them to grow up into tumors. And the reason we're growing them is because we want to figure out ways to kill them more effectively when it comes to patient care. Okay? So once we've grown them, this is the environment that we grow them in. We will oftentimes bring them over to this other room where we have another incubator, just a little, incubator just for the devices. And then we have these microscopes here. And this is a confocal microscope, which basically allows us to take little thin sections, optical slices of the tumors, and understand how they're growing and how they're behaving inside devices like this, so that we can look at the markers and the features of that disease that is different from a tumor that doesn't grow like this, doesn't grow under flow. And that helps us understand what are some of the targets that we can use to attack that disease, and what are some of the reasons that it might be more difficult to treat. And then there's one last thing that makes my program sort of unique from other similar programs. I use light and light-based chemistry, photochemistry, to kill the tumors. And the reason we do that is because the way that this particular approach kills the tumors is different from chemotherapy. It's different from a lot of other targeted therapies that are used. And what we're hoping is that by hitting the tumor from different angles, we can attack the very things that are making that tumor hard to treat. Okay? So we grow them in devices like this. We let them grow into nice tumors. We image them. And then we develop therapies using light to kill that disease, so that we can make it more responsive to chemotherapy and more effective in terms of killing it, okay? And so all I wanted to show you was basically these little LEDs. These are high-powered LEDs. And we shine light on the tumors, so we'll put the device onto the LED. We'll turn this on. It'll be a specific wavelength or a range. And then we have a particular chemical in there that gets activated by that light and kills the disease.
It's fantastic. So I know it's something that our audience is really interested in. Obviously, many of them are undergraduates. Many of them want to participate in research. They might be nervous that they don't have the experience. Everything that you just said is incredibly exciting, and they're going to relate to that in terms of being passionate about it. To what extent do you look for somebody to come in with all of the relevant expertise and hit the ground running in an undergraduate?
Yes, so the main thing I would say is that I was exactly in the same place as you all might be when you're thinking that. When I first entered the lab, I had no experience. It took a great mentor who invested her time in me and taught me what I needed to know. So don't feel that you have to come in as experts. That's what this experience is for. It's for you to get exposed to different kinds of research, to different kinds of mentors, to different people, and to different types of scientific questions, so that you can learn and figure out what it is that you're going to be passionate about, right? You don't go into a lab because the mentor necessarily wants you to become a copy of that individual. You go into a lab because you want to get the tools and you want to start asking the questions that help you figure out what's going to matter to you. That only comes with time and experience.
That's great. Now, as you can see here, one of our students has asked, how did you even come across that photochemistry approach?
Yes, so this was something that actually was observed thousands of years ago, initially by the Egyptians. Then more recently, there was recent advancements in Europe and in America that showed that if you took a particular kind of molecule, called a porphyrin is often what's used, and you gave it light of a specific wavelength that activated that, you could actually trigger photochemistry that would then kill certain types of cells. And what this particular approach, and the advantage of it is there's two big advantages. One, you can actually trigger very specific parts of a cell. So if that molecule localizes somewhere inside the cell, you can actually kill it with light using those organelles to target. And you can spatially confine the light as well as the time that you want to use to kill it in a way that other therapies don't. So how did I come? That's the basis for it. This has been observed now for centuries, and we're harnessing it for the purposes of cancer therapy and other kinds of similar applications. Let me tell you how I came across it.
In 30 seconds.
In 30 seconds, I had a mentor who basically turned me on to this, and she got me excited about it, and now it's turned into my own career. So the one piece of advice that I would give is don't worry about how much you know right now. Find a mentor who's going to get you excited about something and who will teach you, and you'll be just fine going into your own careers.
All right, so that was wonderful. I see our next presenter has joined us. I'm going to log out. Dr. Policek, I'll be right back.
Okay. Dr. Franz, you already log out? Okay. Okay. I think I am able to share my screen anyway, so I'm going to do so. Okay. Can you guys see my slides?
Yes.
Yes? Cool.
You're not in presenter, like in slideshow.
Yeah. There we go. Yes. So actually, Dr. Rizvi's presentation, where he was speaking from is right next to our lab space too. So we share some of the microscopy facilities that Dr. Rizvi uses, some of the microscopes and stuff. Some of our interests are shared. Much of them are quite different though. My group seeks to understand how fluid flow plays a role in tissue development and function. So we're very much interested at a fundamental level of this relationship between cells and fluids in their environment. There's an argument to be made that the definition of a cell is something that segregates one fluid from another fluid. What defines cytosol from the extracellular space? So intrinsic in our understanding of multicellular life is an understanding of fluid transport and fluidic boundaries. So through dynamic tissue movements and when our organs are developing and growing, there are mechanical stresses that build up that drive transport of the fluid from one place to another. As fluid moves, either in our blood vessels, our lungs, but also through our tissues as molecules are brought from the vasculature deeper into tissues, there are forces that are applied by that moving fluid on resident cells. These forces are super important and they can direct processes like morphogenesis, like the shape changes that tissues undergo as they grow, but it's really hard to measure them and it's hard to understand how cells sense them. Do they even sense some of these flows? So the way that our lab approaches this is we build tiny little microfluidic devices that mimic cellular tissues, or I'm sorry, mimic human tissues. So our principal goal is to understand fluid flow in these things, but we also use them to model diseases. So we can get patient cells from our clinical collaborators or hospitals, and we can put them into our devices, and we know how healthy cells assemble into tissue level structure, but we can look at how things are messed up when we use patient cells and use that to screen for drugs and these kinds of things. But central to all of our approaches for both disease modeling and basic understanding is the use of what we call microfluidic technology. This is technology borrowed from the semiconductor industry, and it involves a bunch of strategies to pattern cells and molecules with extremely high resolution. UNC, we're very lucky. UNC, in addition to having a fantastic hospital system, has an amazing world-class facility for microfabrication and the development of microfluidics. So our lab sort of sits in the middle. People in my lab literally walk to the hospital and to the clean room where we do our microfabrication on main campus in a single day. And so, we're looking to merge those things. Undergraduates, I've only been at UNC for a couple of years, but in those few years, I think we've had 9 undergraduates pass through. And what the undergraduates work on depends on the specific interests of the individual and maybe strengths that you're not even aware of until you start working in the lab. So the way I approach undergraduate research is I tend to give each undergraduate a couple of options and see what they're interested in. Give them a little bit of free rein for maybe the first month in the lab. And then we discuss a project that's of interest to our lab and to the undergraduate, where we can define specific milestones and objectives. Because it is kind of hard to culture some of these cells and to learn that process while you also have classes and everything going on, the projects within my lab for undergraduates tend to focus a little bit more on either the development of technology or computational methods. One of the things my lab does, puts a lot of effort into, is building the three-dimensional environment of tissues ex vivo, and that makes some analysis pretty tricky. It's hard to figure out if cells move, where exactly they're moving, or how cells and other things are reacting in three-dimensional space. So we do a lot of computational simulations to sort of make sense of our data, and that's a great project for undergraduates. But some of the undergrads I have had in the lab started out on projects like these, like building pumps or writing computational code, and have gotten so interested and spent enough time around the lab that they have learned cell culture on their own and started to build their own systems. That's the dream for me, is that we can train you on all the other aspects of the research in the lab, but it's a little bit hard to do with all the other things you have going on sometimes.
So Bill, I apologize for missing the first part of that, but I was able to connect pretty quickly, and I have to say I've been, especially you, Dr. Imran, really impressing the students just with the description of your work, the impact of the work, opportunities for undergraduates. I have to say, I'm realizing I put all this time into organizing this, and then I sat down in my own lab. I'm like, oh, I have to talk about my own stuff. Now what am I going to do? But I'm that you're getting the chance to do this. I wonder if you could speak a little bit to, you mentioned tech development and you mentioned computational tools. Those are efforts that often can be done without, needing to worry about 6 feet social distancing. And, within the pandemic, a lot of our students are really wanting to hear about opportunities that they might have available despite there being a pandemic that is sort of limiting a lot of things on campus. So in your lab, have you found ways to pivot for students to find success where they don't have to be, within the confines of a single room and inside the lab?
Yeah, that's a fantastic question. I mean, and I'll be totally honest, it's a learning experience for us as well. Our sort of how I motivated the computational approaches is very much how we thought of it before, that largely we use them for analysis. So it's a little bit hard to advance the computational goals without getting new experimental data. However, we have started to, there's a lot of inertia in some of these projects, but over the course of the summer, as we've realized this is going to be with us for a while, we need to restructure how we approach some of our problems. We've spun some of our analytical approaches into predictive approaches. So we are trying to take limited data sets that we have already in hand that we could send to an undergraduate, for example, and then build models that might allow us to predict what would happen if we change certain variables, with the goal of eventually being able to run the experiments. But even if we can't run the experiments, some of those predictive models might be able to tell us, this is where you should spend your time, this isn't worth your time, that kind of thing. So, it is a work in progress, but we have figured out ways, particularly when it comes to computation, to do things remotely.
I think it sounds like your philosophy on computational modeling is very much similar to my own, where we're not a modeling lab. We'll model when we don't know what the experiment should be and we need to figure it out, or we've made a measurement and we have no idea what caused the difference that we measured, and we need to figure that out. And so that's really good. If anyone has, we have maybe 2 minutes here. Will you be teaching bio-inspired engineering again, Dr. Polachek? That was one question.
Yes, I will be. I hope I will be. I mean, that is the plan. Yeah, it'll be offered each fall.
Good to know.
So next fall, yeah.
The Kirti looks like she's already enrolled. that you might have students for Dr. Polachek while we have him here. My next log off and coming back on, it's going to be longer than the others. We've been in Mary Ellen Jones so far, but I'm going to, for the next 3, be moving around campus actually to go to different buildings on your behalf.
Any other questions?
I will say that I've told everyone that I'm going to follow up with a list of the PI names, the laboratory websites. Those usually contain contacts, emails. I've told the students that our faculty are really wonderful and most welcome emails from students, though they shouldn't be surprised if, you know, it takes us a little bit of time to get back to them. We're always welcoming those connections.
Absolutely. Do you, would it be helpful to you if I elaborate on that a little bit as you transition to the next place?
Yeah, I have to go over to Dr. Diekman's lab. So if you are kind enough to stay on, I'm going to take 10 minutes and I'm going to walk over there. Thanks. Really appreciate it. I'll see everybody soon.
So yeah, I get to bore you guys with some more of the details of our work. I actually, this was a presentation full of slides, and I just, as so often is the case, I just talked through it instead of actually showing you guys the stuff. So I'll show that in a second, but I do want to elaborate a little bit on what Jason was saying about how to get in touch with faculty you're interested in doing research for. So for me, I'm super lucky because the BME department, we get amazing undergraduates, but I also get interest from other departments around campus, particularly in mathematics and the physical sciences. So the emails that jump out to me are ones where I can tell the person is interested in specifically what we do. So scratch a little bit below the surface. I'm not expecting you prospective undergraduates to go read every paper we ever published. But in research, papers are the currency by which labs judge their own success, and they are the track record of what has actually been accomplished and what the community has agreed upon is an accomplishment. So if in an email you say to me, I saw this paper, I don't really understand the details because I'm new to this, but it seemed really interesting for X, Y, and Z, that tells me you went a little bit beyond just checking out the website, and I'm immediately going to follow up with a discussion. So for me, that's like a key threshold. I will interview students who don't say that, but to me, that's like the clearest way to show real interest in what the group is doing. The other thing I would say to those of you interested in doing research that I wish I knew when I was looking for labs is each lab is like its own world. There's totally different cultures. They operate in different ways. In some labs, you might be assigned a graduate student mentor who handles your day-to-day. In other labs, you might interact directly with the professor. There are benefits and drawbacks to both. Some labs are, people are just in there to do work and then they go home. Obviously, this is a little different in COVID, but in other labs, they're very social and people tend to hang out, you know, into the evening and stuff. And what I would say is, you guys are all young, just starting out on your research career. There's going to be some flexibility in what you find interesting. And you want to find not just the subject that interests you, but a culture where it brings out the best in you. If you're interested in doing research, I imagine you could be interested in working on a few different types of problems, but you're not going to make much progress unless you really want to show up to the lab. Because it's kind of, the thing about research is there's a lot of work that you kind of don't get credit for because most of the things you'll try don't work. So you got to really love what you're doing in order to make a real impact. So if you're interested in a lab, it's of your benefit to meet not just the PI, but maybe somebody who's actively in the lab too. And, you know, for at least for me, if the student says, you know, I'm interested in your lab, could we speak? And then could I talk to somebody in your lab? I'm more than happy to make that connection, especially these days. My poor graduate students, all the poor graduate students are holed up, not seeing new people ever. So I think they'd be really happy to meet some of the undergraduates, at least for us. The undergraduates are usually a source of fun when things are challenging in the lab. Do you guys have any other questions? No? Okay. I hate to bore you with research until Jason's back on. So I'm also in both the process of teaching and also grant writing right now. And a part of the grant that I'm writing is like trying to introduce the thought of mechanical forces in fundamental biology classes. So I'm primed to do a little preaching to you guys. So one thing that I mentioned, our lab is really interested in fluid forces, but we come from a standpoint that forces are just as important as soluble signals. So in your biology classes, when you may have learned about transcription, translation, the central dogma, and how cells take genetic information and turn it into proteins, at every stage, there are mechanical forces involved. And I can point to specific examples at every stage in the cell, from transcription to where cells are sitting in the microenvironment, in which the application or removal of mechanical force can lead to a different biological outcome. So for example, when mRNA is being translated into protein on ribosomes, there is a force that needs to pull the mRNA through the ribosome in order to assemble amino acid chains. There are certain diseases in which cells can't generate sufficient force that leads to mutated proteins, and that can cause things like cancer. So at the very fundamental level, forces are involved. The problem is these forces are tiny, really, really tiny. You can imagine that if there are millions, billions, I don't know the number, trillions of pieces of mRNA that are being pulled through ribosomes in your body at all times, and you obviously don't feel it. These things are really tiny. Some of the length scale comparisons are amazing. You know, we are trying to measure forces that if you compare like the mass of a human to the forces we're trying to measure, it would be the same as the height of a human to the distance of the moon. So the ratios are about the same. And what's kind of remarkable about biological systems is that they have, I mean, we can generate forces that are really high, right? We can bend metals, and we can lift heavy things, and we can push stuff. But the tissues and the structures that comprise the tissues also generate forces at really tiny length scales. So it's a pretty amazing, you like imagine, you know, thinking about designing one of these systems from scratch. You got 5 gears in your car that can apply different magnitudes of torque, but we're not talking about the 9 or 10 orders of magnitude difference that exists within living systems. So one of our overall goals is not just to understand forces in biology, but to try and this kind of links into the class that it's a new class, but I'm developing for the fall, but to try and take inspiration from how naturally evolved systems have come to use force to propagate life, and to try and use those principles to better our engineering approaches to generating force for, you know, whatever we want to engineer for. So, so yeah, that was not super coherent, but force is important. That's all. Yeah, and yeah, I hope you remember that. Anyway, no questions? Somebody's got to have a question.
What will the class in the fall that you're developing be called?
Bio-inspired engineering. Yeah. Someone asked a question about that. Yeah. So I also teach a junior and senior level biotransport class. That's more like traditional engineering, math, and stuff. But then the class in the fall is design focused. Is actually supposed to be a prototype deliverable at the end, but obviously COVID got in the way, so we're doing finite element models instead. But still, it will be only better once we can get back into maker spaces and stuff.
I have a quick question. I'm an NC State-based student, sophomore, and I was wondering, like, takes place at Chapel. Is there like any online research opportunities related to this that I could possibly take part in from the NC State side?
Yeah, if I may chime in. So Dr. Michael Danielle and Dr. Ashley Brown are co-hosting an event that's going to be identical to this in spirit, but would be from NC State. Another thing I would add, and so they haven't sent out the announcements, but it should be coming up soon. Another thing I'll add is I should have said this up front, and Dr. Polachek, I'm sure, can attest to this as well. You know, I'm so glad we have NC State students here because we are, or I shouldn't say NC State students, NC State-based students, because we're a joint program. And we've had, at least in my lab, you know, 6 or 7 undergraduates from NC State come up and be successful and participate. And I know that a lot of our UNC-based students go down to NC State and do the same. So under normal circumstances, we have a bus that goes back and forth. Those are really wonderful opportunities. So thanks so much. And I'll be sure to follow up with Dr. Danielle and Dr. Brown about when that event might be. Good question. Dr. Polachek, thank you so much for filling in as DJ here for a moment. I'm over in Dr. Scott Magnus's lab. And so I'm going to go ahead and continue our tour.
All right. I'm going to sign off. It was nice seeing all of you from afar. And please don't hesitate to send me emails if you have any questions. Thanks for putting this together, Jason.
You got it. Thanks.
Hi, everybody. My name is Miriam Ock. I am a 3rd year in the biomedical engineering department. I'm a grad student, and I'm also an MD PhD student based at UNC. So I did 2 years of med school, and now I'm taking a leave of absence from medical school to do my PhD. And so if anyone ever wants to talk more about that, I'm happy to chat. But here we are in the Magnus lab. It's definitely a very interdisciplinary lab. Scott Magnus has joint positions with cell biology and physiology, with biomedical engineering department. We also work with a lot of cores that are kind of integrated within the lab. So the CGIBD, the Center for GI Biology and Diseases, and also the Advanced Analytics core. So there are a lot of interdisciplinary and collaborative opportunities, which is always a lot of fun. And so we can kind of maybe take a look at a poster over here. We have a whole slew of posters, but basically the gist of it is that the Magnus Lab is really interested in studying a human and mouse, but in particular intestinal stem cell fate. So we do a lot of cool biomolecular assays and biomolecular studies to be able to actually better visualize these different cell types through fluorescent microscopy techniques. So we can actually use CRISPR-Cas9, if y'all are familiar with that, to basically introduce fluorescent markers that are associated with different markers of either stemness or differentiated levels of cells. So essentially, like if you have a stem cell under the microscope, it'll glow green. And if it differentiates into a goblet cell, it might turn blue. So basically, with those kinds of tools, we're hoping to better be able to visualize and track cells both in various mouse models, so we can do it sort of in vivo, and as well as in vitro through various engineered platforms, since we do have a lot of engineering students. And so it's a lot of really cool technologies. Let me see if we have an example. For another example is like this micro-raft array technology that we developed with our collaborators in the Albritton lab, which they've now moved to the University of Washington, but we're still very close collaborators. Yeah.
No,
feel free, go ahead. And basically this is like a tool that they had developed and we use where you can actually grow single cells on individual little punchable platforms, essentially like a little punch box, and use a microneedle once you select a cell of interest based off of its morphology or its fluorescence patterns to like kind of punch them out and then transfer them into using a magnet to be able to transfer them into a single well and be able to like grow cells clonally from that single cell. So this is just like an example of a cool technology that our lab uses. And I'm gonna take us over a little bit more, a little farther to, let's see. This is actually a poster from Christina Rivera, who actually just graduated this year from the BME department with her PhD. And she actually developed a platform to be able to study hypoxia with human intestinal stem cells. So she actually worked with, was a dual student, so she had a co-mentor over at NC State, where she would make the devices and then bring them over here and basically be able to, in a very tunable and controllable way, control the oxygen levels that her cells were exposed to.
and basically be able to see how different levels of oxygen affected the cell's behavior and affected kind of like their growth, and would kind of plate them into organoids, if you've ever heard of those or worked with those, and see how it affected how large they grew and how well they grew and that kind of thing. So that's just a different example of the kind of technology. But enough about posters. I'm going to go ahead. Hey, Jarrett, we're doing our virtual tour. The grad students in their element. So this is our lab space. We have a lot of bench space. Recently, we've kind of acquired more bench space as we've had people who were sharing the lab space with leave. So it's kind of nice. It's been useful during COVID when we needed the space. But basically, like over here, we have kind of your standard like gel electrophoresis setup and your UV imaging system, so that we do a lot of cloning in the lab. We have to be able to actually extract and put together different fragments like puzzle pieces to be able to design these fluorescent cells, sort of fluorescent biosensors. So like this is where a lot of the magic happens, where we actually run the gel and extract the bands of interest and purify them and whatnot. So that's, I guess, I don't know, this is maybe interesting. So this is actually one of our actual tissue slicers. So a lot of times people will take samples to a tissue core and have somebody do that for them, but that's actually one nice thing about our lab is we're pretty self-sufficient. So if we want, we can actually freeze down bits of tissue and then be able to actually slice them up ourselves and put them on a slide and visualize them, which is really useful and efficient if we want to be able to do things quickly or have a particular orientation that we want to keep the tissue in. So that is one tool that's available here. I'll kind of walk us along the hallway we were in before, where we have sort of like several spaces that are technically our lab space or shared with the cores. This is the poster hallway. I'm taking us to the tissue culture space. One of them, this is Willow, one of our awesome lab techs. She's working diligently. So basically we have two hoods here right now during COVID. We're only trying to use one at a time, but we can do your standard tissue culture. We work with like standard 6-well plates. We also work with trans wells, so you can kind of change what you add to the bottom versus the top, depending on your needs. And mostly we work with either collagen or matrigel, extracellular matrix, as our substrate of interest to grow cells on. And that seems to be working pretty well for being able to maintain human intestinal stem cells. And then, I don't know if Joseph is here. Joseph, hello. Do you mind if we show some cool images to the undergrads? If you want, you can go ahead. I think Joseph is in using the microscope. You want to show them what you're looking at?
Pull up something good. Right now I'm looking at bad stuff.
This is Joseph, our postdoc.
Hello, Joseph.
Say hi to the BME undergrads.
Hi, undergrads.
Oh, they can't see the screen at all, even if I pull something.
If I move it closer, I think they will be able to.
Why is this bad?
So Joseph is working on some pretty cool models that we call PIP-Fucci, which actually allows us to see cells in different phases of the cell cycle, and they will turn red or green or kind of a combination of both, depending on what phase of the cell cycle they're in.
Yep.
Yes. Cool stuff.
Yeah, it's working on monolayers here. It's,
yeah.
Have you told them about monolayers? Pretty much it's a system our lab uses and not much of anyone else in the nation, and it's cool.
Yeah. So basically, yes, Joseph kind of summed it up nicely. But the cool, the cool thing about our lab and our collaborators is that we were able to develop a way to be able to grow like healthy cells in a sheet or an organoids, which is really challenging. You know, cancer cells will kind of grow on anything. They just need like something to stick on and a little bit of food. But like healthy cells, if you want to actually understand normal physiology, is a little bit trickier. Cells need a little bit more nurturing, a little more high maintenance, like me. But I'll say, actually, Joseph is currently the only person in the lab who sort of has an undergrad that he works with. But Anna, I don't know if she, you had a chance to meet her at the panel, but she's been a wonderful help with the lab, and she's also kind of has her own independent cloning project. So she's working with Joseph to kind of be able to develop some of these systems on her own. And I guess we can't go in this other room, but this is actually, they're probably doing a FACS experiment. So fluorescence activated cell sorting, we are one of the few places that actually do human intestinal stem cell FACS
which is really cool because you can actually study individual cells. You can actually divide them and gate them based off of the levels of intensity of their fluorescence. And then actually downstream, once you have these different gates and different groups, you can actually do further processing and do quantitative PCR and basically see how different levels of intensity correspond to different gene expression and look at other kinds of downstream markers of interest. So that was a lot.
It was great though.
I think I covered most of it. There are some people in the lab who actually work with gastric tissue too, so not just intestine, but some people work with the stomach. And it's kind of a mix of just, other people are working on bile acids, other people are working on various toxins. So it's a little mishmash, but we have fun here. It's a good group, and I'm happy to take any questions.
Yeah, I was going to say, with the little bit of time we have left, that was awesome. You said one undergraduate's working, and we had a really pleasant experience with her. She gave great advice on the panel. Are there opportunities for BME undergraduates this coming year, especially amid COVID?
Yeah, so I think there are always ways that we can kind of discuss and work around based on people's interests. I think it depends on whether or not the project would have to use a lot of the other core facilities that have more restrictions. Like, for example, my project, I do a lot of work over at Channel, which is the Chapel Hill Analytical and Nanofabrication Laboratory. And right now it's a little bit tricky to do training to use those to make devices because they have the restrictions for one person in the room at a given time. But there are always other projects and cloning and things like that that are easier to do in-house. That if people are interested, we can always find ways to make it work and make it a mutually beneficial experience. So yeah, if you have any questions, you can always talk to me. I don't know if you send out everybody's emails and or Scott, and I'm sure there's always a way to work something in if there's interest.
All right. Well, thanks for sticking with me, everybody. We've got a couple more spots. I'm going to be gone for the next 10 minutes or so, but keep with us. We've only got 2 more, and you don't want to miss them. Here's your commercial break. All right. Thank you for still sticking around here. You can hear me? I'm glad my Wi-Fi in all these buildings is going well. All right, we have 2 more labs to go. I'm going to turn it over to our esteemed colleague here, Dr. Brian Deacon.
Hi, everybody. Thanks for joining the Zoom here. So what we do in my lab, we have 2 main projects. So I'll probably spend most of the time on some of our genome editing work that we're doing, just describing how that process works, because things like CRISPR and the powerful techniques you can do around genome editing have gotten a lot of press and are certainly taking the research world by storm. So you guys might be interested in that. The other project we're doing is more on cellular senescence and aging. So my lab studies cartilage and osteoarthritis. And basically we're trying to figure out why so many people, as they get older, why their cartilage starts to break down and start having symptoms of osteoarthritis. So it's commonly thought of as like a wear and tear disease, more mechanical approach, and Dr. Franz would probably take that approach. But those of us more on the biology side are also studying how the cells change with age and how they actually will respond differently to the same loading conditions when the cells have been around a lot longer. So if you're interested in that, definitely send me an email and we can have that discussion. But for the genome editing, what we're trying to do there is understand the genetic risk factors for osteoarthritis. So about half of the risk for getting osteoarthritis is genetic. So people that do twin studies and all these other things to figure out family risk. But it's not just 1 or 2 genes, like some other diseases where people have identified these strong risk factors. Osteoarthritis, all of the particular genes that people have found have a very small contribution to the overall risk. And so it's been really hard to figure out why these different changes in the genetic code would result in more osteoarthritis. And so what we're doing with genome editing is we can take advantage that we can take chondrocytes from cadaveric tissue donors, so people that have passed away and have donated tissue through a bank so that people can do research. And we can take those cells from individual people and go in and tinker with the genome and make just 1 single base pair change, or make a bigger change of deleting a whole gene. But basically we can change just what we want to in the cell and leave everything else exactly the same. And so I'll kind of talk you through the different process for doing that. But the reason that's so powerful is to figure out why genetics drive osteoarthritis. Normally you need like 50,000 or 100,000 patients in what are called genome-wide association studies, or GWAS. And you have to do that because there are so many lifestyle factors, so many differences throughout the genetic code between people, that to find whether one particular base pair is going to have an effect on OA, then you need to have so many people to kind of get through the noise of the rest of it. But if we can get rid of all that noise and only change out one base pair in these cells, then we have a much better chance of seeing what that does. So we work with a lot of people. This is the Thurston Arthritis Research Center is where we work. That's why Jason had to walk all across campus to come to our building. But we have a lot of great collaborators that work in the same areas, and so that's a lot of fun. And we're really just right across the plaza from most of the BME folks. So what we would do is, first we would get the tissue donor in, and I guess I can show you the cell culture room. And I didn't know the video was going to be so good. I'll even show you a chondrocyte, okay? Chondrocytes are the only cells that are in cartilage, and so that's kind of nice too, as opposed to these complicated tissues. So this is our incubator, and this is good because we could not all fit in this room, so we're taking advantage of the video. This is just the media that keeps the chondrocytes going. This scope actually has a display, so you'll hopefully be able to see it. All right. Let's see. All right. So right here, there's one chondrocyte, another chondrocyte, another chondrocyte, another chondrocyte. All right. This would be like this one. You can kind of see the nucleus. You have all these speckles in the cytoplasm, and then this is the nucleus right there in the middle. All right. So we get those. We let them recover from being isolated. You know, these cells were in somebody's cartilage like 3 days ago, right? So they've gone through a lot to get to this point. So then we will take them, and I'll show you some of the other equipment. We'll take them, and after they've recovered, then we deliver the CRISPR-Cas9 components, which actually we have a cool poster in the hall while we're at it. Okay, so the way CRISPR-Cas9 works is there's a protein and enzyme called Cas9. That's what actually cuts DNA, but you have to complex with it a guide RNA, and so that's what is going to tell the Cas9 where to go and where to cut very specifically in the genome. All right, and so you do that, and that will take this protein right to the exact site and cause a break. And the cell hates double-strand breaks. It's like the worst possible thing for a cell. And so it's going to do whatever it can to fix that break, including if you provide some template DNA, then it's just going to shove that new DNA that's floating around to plug the hole. And so that's what we take advantage, like for this cell, for example, that had a T at one particular base pair and both copies had a T, that we can send a repair oligo that is an exact match to the DNA that's there, but instead it's going to have a C right at that one place. Okay, and then the hard part of doing this is we have to screen all these different cells to find the few that actually do this correctly. And so that's where we do PCR. So I'll just show you a thermal cycler, which is like a workhorse of molecular biology techniques. And so it's basically amplifying a small segment of that DNA over and over again. Okay, so these machines, so they'll cycle through and go up to 95 degrees to break the DNA apart, and then they'll cool and come back together and amplify. And so then we can take those PCR products and we'll take them over to the to run an agarose gel. This is something that maybe you guys have seen or done in a lab, where we take those products and we'll have like a little hydrogel there, put the solution in, turn on the voltage, it'll run down. We can see how big those fragments are. And then we can screen that way. And once we find the colonies of cells, the particular cells that have been changed, then we could expand those up and make our little pieces of engineered cartilage so that we can study it. So that's all that I've got. Is there time for questions?
Yeah, so I would really be interested. I mean, you're doing fantastic work, and it's totally innovative within the scope of other labs within biomedical engineering, largely because of the collaborations that you have with Thurston. I know that a lot of our undergraduates are interested in cutting edge technology. Can you speak to the role that undergraduates have played in your lab, and especially amid COVID, when they might be feeling that maybe the opportunities that they would have otherwise have aren't there, just putting their mind at ease a little bit?
Yeah, it's certainly, you know, we're not a computational lab, so there are some things that we definitely need to be doing hands-on. So it's been a challenge for all of us to navigate that. But we have been doing things like when we sequence the cells to figure out if they've had the successful edit, you know, that can be done remotely. And so getting people to kind of help interpret. But we have had a good number we've had 4 different undergrads. We've had 2 from UNC and 2 from NC State that have come over to work with us over the last couple of years. And 2 of them were able to stay over summers. And so especially for these kind of techniques that take you a few tries to get it down and kind of long-term projects, that's something that's been really effective for our undergrads, is if they can be a part of a lab kind of early on in their careers and just understand it, kind of make a good impression on the fellow lab mates and everything, and then look for an opportunity to spend a whole summer doing research. And so we've had some students have some success there and get on papers and things like that.
Yeah, I like that. 2 reasons. One, we know we have some NC State students that are joining us today. The other is you were talking about long-term, the fact that it takes a semester or more to learn a technique. Other people have said, you know, you're going to learn a lot of the skills that you need on the job, but you know, after you've learned them, it takes a while to put them into practice. And the goal for all of you that are joining us, it should be more than on your CV a line item that says I was in the Diekman lab from July to June with some dates. Right. What you're looking for is deliverables that you can speak to show that you learned some skills, whether that's an abstract, whether that's a co-authorship on a paper. And I think a lot of the faculty really do value undergraduate contributions in the science to that level.
For sure.
Cool. Well, thank you again. I've got one more lab to show you. This one's the furthest away. I need 15 minutes to walk over there. And so here is your bio break. Maybe put your mac and cheese on the stove so you can have dinner right after. I'll be back soon. Switch it over to our last lab, and that is Dr. Andrea Giovannucci's lab. And we are in the Keenan Laboratories building.
All right. Hello, everybody. My name is Jake. I'm a postdoc in the Giovannucci lab. He can't be here today because he and his wife just had a baby. So congratulations for him. So I'm the one who primarily uses this rig anyway, so it'd be good for me to give you a chance to look over our experimental setup and explain to you a little bit about what it is we actually do in this lab. So this lab is sort of a combination of neuroscience and machine learning AI techniques. So that takes place as far as we're in our interpretation of the neuroscience, looking at how it relates to machine learning algorithms, and also applying machine learning algorithms to actually analyzing the data. So the primary method of data collection which we use is a combined 2-photon and optogenetic microscope. So here you see a little wheel. We could have a mouse set up on this wheel, and it goes underneath this microscope. And then this microscope is actually 2 things in one. So it can monitor neural activity with single cell resolution by using a special genetically engineered virus that makes neurons glow. So we can take videos of neurons glowing basically with GCamp, or with a different virus, and at the same time in the same mouse, we can target specific points of light to target specific cells and activate them. So we can read and write neural activity, right? And the way they do that is actually to use deep learning algorithms to, on the fly, online, create holograms of light within the mouse's brain. So if you think of a hologram, you could think of like the old Star Wars movies where you see Princess Leia. She's kind of like a figure of light emitted out of R2-D2. That may be a little dated reference. But that's basically what it is, except instead of Princess Leia, we have little points of light that correspond to cell bodies, right? And we want to target those cell bodies, activate those cells, and see what that very specific subgroup of cells does in relation to the animal's behavior, right? And so this is our behavioral setup where the animal would go. So we have This objective is for monitoring neuronal activity and delivering the stimulation patterns. We also have these other behavioral cameras. And so we just have one set up right now, but in later stages, we will have 2 to 3. And what that will do is using algorithms, an algorithm called Deep Lab Cut, we'll be able to track the animal's movements in 3 dimensions. So then we can relate activity and movement related portions of the brain to actual behavioral movements. And let's see, so undergrads in the lab, we have a few different roles which that can take. So we have experimental, more biological hands-on projects. So that could involve training animals. That could involve more biological processes like looking at the brain tissue and slicing it and doing histology and that sort of thing. We have more engineering-oriented projects. So you see all sorts of rigs here set up to hold the animals. We have these rigs to hold the behavioral cameras, and all these systems have to be integrated. You can see here, we have this setup. All of these computers are to run various things that happen on this rig. I don't know if the lighting is good, but we have a whole bunch of electronics, and I think we have 3 computers on this rig to run the microscope, to run the behavior, and then I think we have 2 for the behavior. We have 2 computers for the behavior. And so getting all these systems to work together and to build the necessary components takes a lot of engineering know-how. So that's something you could do. And then last is we have a lot of software and data analysis opportunities. So this type of rig is capable of generating immense amounts of data. It's gigabytes per experiment. And so you need robust data analysis pipelines in order to analyze that. You also need you need to look at it a lot of different ways, right? You'll get a very complicated data set and you have to turn it over multiple times. So those are the 3 roles that we've had people take on in the lab. We've had, I think, 10 undergrads from BME in this lab across all 3 of those roles. And they've so far worked out really well. I really like working with them. And so those are the big opportunities. We're still kind of getting things up and going and recovered after COVID, but we kind of got things hammered out as far as our protocols for staying safe and for being in the lab. And of course, we also have a ton of work from home opportunities since we do a lot of software and data analysis. So I think that's everything that I can get at a first pass.
That was awesome. So I want to make sure that we have opportunities for questions. As you know, every time I log out and log back in, the chat clears itself. So if you've asked questions before, maybe Becky could chime in and just read them out. Otherwise, remember this is Dr. Andrea Giovannucci's lab, and he joined the department only about 2 years ago, and the extent to which he's been able to set up and hire wonderful people like Jake to get the research going. And I'm looking around the room. It is fantastic looking. It's so fun. I told him I want to come back and geek out a little bit on my own time. Do you have any questions?
Jason, there aren't any questions in the chat.
Thanks, Becky. Does anyone have any last minute questions about our day? Just to sort of reiterate, I'm going to be following up by email with a list of each lab that we went to, their lab website, you can check it out. That should have contact information on it. Something you want to look for, again, is a lot of these lab websites will have expectations for undergraduates and some information about what the PIs are expecting if you want to email them about research opportunities, right? Show that you've given some thought to the research. Don't send an email that says, I want to do undergraduate research, period. Call me, right? You need to show that you've given a deep dive into their website. Maybe you've read a paper. Read a paper and say, you know, when I looked at your paper, you found that thing you didn't expect. I was also surprised by that. Can we meet to talk more? I'm at UNC, and I'm looking for undergraduate research opportunities. but I don't just want an opportunity to add something to my CV. Your research is the research I want to get involved with, right? So give it some thought. I'm killing time now, folks, while I look for questions in the chat. If there aren't going to be any, if you have any follow-up questions, you can send them to me. I'm happy to field your questions. But in the meantime, thanks so much for spending the afternoon with me. Thanks to Jake again and Giovannucci Lab. And go have that mac and cheese you put on the stove at our last break. Thanks, everyone.